Zeddy, this is my teaching presentation for Anatomy and Physiology 201 lecture in the subject of cellular respiration and glycolysis. Let's start with the definition for cellular respiration. It is a set of metabolic reactions and processes that take place in the cells of an organism to convert biochemical energy from nutrients into adenosine triphosphate, otherwise known as ATP, and then release waste products. There are three types of cellular respiration: glycolysis, Krebs cycle, otherwise known as the citric acid cycle, or electron transport chain. Today we will be reviewing glycolysis. The type of cellular respiration that we will be reviewing today is glycolysis, which is defined as a chemical process that occurs in the cytoplasm of a cell, in which glucose is broken down into pyruvate and gains 2 ATP and 2 NADH. Glycolysis in 10 steps. Step 1. You begin with the molecule glucose, an enzyme called hexokinase, and ATP. ATP will donate a phosphate to the glucose due to the enzyme hexokinase, which creates a new molecule called glucose 6-phosphate. Once we have the molecule of glucose 6-phosphate, it is acted on by an enzyme called phosphohexoisomerase. This enzyme rearranges the glucose 6-phosphate into fructose 6-phosphate. Step 3. With the introduction of another enzyme called phosphofructose kinase 1, this enzyme attaches another phosphate group from another ATP to our molecule, which then creates a new molecule called fructose 1,6-biphosphate. In step 4, fructose 1,6-bisphosphate is split by an enzyme called fructose bisphosphate aldolase. Aldolase is designed to break carbon-to-carbon bonds, so you now have two molecules, one called dihydroxyl acetone phosphate and the other glycerol dihyd 3-phosphate. In step 5, it is conducted by another isomerase called triose phosphate isomerase. This converts the dihydroacetone phosphate into glycerol dihyd 3-phosphate. So there you will have two glycerol dihyd 3-phosphate molecules after step 5. In step 6, we meet glycerol dihydride 3-phosphate dehydrogenase, which is responsible for moving a hydride ion onto an electron receptor such as NAD+, which will give us NADH, which gives us the molecule 1,3-biphosphoglycerate. In step 7, we introduce phosphoglycerate kinase to the molecule, which will release 2 ATP. And will produce 3-phosphoglycerate. Starting on step 8, we introduce phosphoglycerate mutase, which will create the molecule 2-phosphoglycerate from 3-phosphoglycerate. Step 9, enolase is introduced which creates a double bond by removing the hydroxyl group, which leaves us with phosphoenolpyruvate. And finally, for the 10th and final step, we need the final enzyme called pyruvate kinase. This enzyme is going to remove the phosphate groups from the molecule, attaching them to ADP, creating ATP. Remember, there are two molecules since the split in stage 4 from the aldolase and the intro of the tyrosine phosphate isomerase converted dihydroxyacetone phosphate into glyceraldehyde 3 phosphate. So finally we are left with pyruvate, which will be the substrate needed for the Krebs cycle. Thank you for watching this presentation, and hopefully it didn't bore you too much.